In science, ideas, conclusions, and even theories should be continually tested. And so, it is appropriate to question and to test the evolution model, just as it is to test and question alternative models like the creation model and the design model. They do, after all, make different predictions. So, for example, if we were to study a gene for a hormone like oxytocin, the evolution model predicts that human genes should be similar to those of other organisms. These similarities should reflect a nested hierarchy of relationships, and there should be intermediate complexity in that other organisms should have some, but not all of the features found in humans. In the creation model, however, there would be no necessary similarities required in organisms which are 100% unrelated to each other. Even if there were similarities, such as all organisms or all vertebrates possessed a certain hormone, there wouldn't certainly be any requirement for a pattern of similarities. The major tenet of intelligent design is that of irreducible complexity. There should be no organisms which would have some, but not all, of the complex features found in humans. These models are testable. Given that hormones are simply signals, they do not have to have a specific structure as long as there is a receptor on target cells which recognizes the signal. So, are there similar signals to oxytocin in other organisms? There are, even in invertebrates. These similarities are in the length of the signal, the sequence of amino acids in the signal, the position of the cysteine residues, which will form the disulfide bond affecting three-dimensional structure, the cleavage from a precursor protein, which I'll get back to, the intron and exon organization, the effects on systems of the body, such as reproductive systems, the ability to affect behavior. All of these are similarities that the evolution model predicts and other models don't. As predicted in the evolution model, and opposite of what is predicted in the intelligent design model, oxytocin signaling shows intermediate levels of complexity in different organisms. So while this is the signal in mammals for orgasm, for the uterine contractions during live birth, and for the ejection of milk in mammals. Nevertheless, homologs of this signal are found in much simpler non-mammalian organisms, even in vertebrates. And oxytocin is part of a gene family, like so much of genetic diversity. Its origin lay in the duplication of existing genes, forming a gene family, rather than a sudden appearance of a unique gene. There are many other homologies in oxytocin signaling which reflect humans' relationship with other organisms instead of showing that humans are 100% unrelated to them. And so, like in humans, in other vertebrates, oxytocin is synthesized in the hypothalamus, but axons travel from the hypothalamus through the infundibulum to the posterior pituitary, where they are released. And oxytocin is not produced by itself. It is made as part of a precursor protein, which in addition to oxytocin includes the amino acid sequence for neurophysin 1. And this precursor protein is then cleaved to produce oxytocin and neurophysin 1 as separate signals. There is no reason that this feature has to be shared in organisms which are 100% unrelated. Even if organisms all possessed the same gene, they could possess the same copy of it. So it might appear that the gene kind of appeared from nowhere and was consistently present in organisms. But what we observe is a nested hierarchy of relationships. Not only are humans related to other organisms, but to varying degrees, most closely related to apes, and then other catarrhine primates, and then other primates, mammals, etc. The sequence comparisons of the oxytocin neurophysin gene is consistent with what we observe from other genetic comparisons. So human sequences are virtually identical to other apes, so 98% identical to that of chimps, almost 98% to the sequence of gorillas. And so it does not seem that humans are 100% unrelated to these organisms. They are almost 100% identical at the sequence level of genes. All of the sequences, which are more than 95% identical to the human sequence, are those of catarrhine primates, which include the apes and the old world monkeys, just as is predicted in the nested hierarchy of relationships that one gets from anatomical studies or different genetic studies. And then other mammals, such as the horse sequence, are similar to a lesser degree, and so forth. And so the study of oxytocin and neurophysin overwhelmingly support the predictions of the evolution model and overwhelmingly refute the predictions of the creation and intelligent design models.